Welcome all. Now in this session we'll discuss about the structure of the chromosome. Okay, so this is the figure showing a chromosome. Okay, so in the center there is a centromere and each chromosome has got chromatid. Okay, this is one chromatid, this is another chromatid. And this chromatid in the center there will be centromere and it will have one short arm and one long arm. Okay, and the ends of the these sister chromatids, they are called as telomeres. Okay, this is also a question we have seen last time in our session, importance of telomeres. Okay, so that we have discussed. Now chromosomes are thread-like structures. They are made up of DNA and proteins, that is histones. The function is that they carry the genetic information, that is gene. Okay. So structure of the chromosome, it has got a chromatid, that is one half of each chromosome. It contains the DNA. Then there is a centromere, that is the constricted region. That is the site where the spindles will attach. Okay, attachment of the spindle happens here. Then at the end there are telomeres. These are the ends of the chromosome and they protect the DNA. Then there is a short arm and the long arm. So P arm is labeled as a short arm, or short arm is labeled as P arm, and there are certain genes which are located here. There you have a long arm which is also called as Q arm. There are certain genes which are located here also. Okay, so total chromosomes in the human are 46. Functional, there are two types of classification. One is a functional classification. These are autosomes and sex chromosomes. Okay, and then there is a structural classification, that is centromere based, depending upon the location of the centromere in the structure of the chromosome. Again, these chromosomes are further classified. Okay, so we have 46 sets of chromosomes, and functionally we have divided into autosomes and the sex chromosomes. So 46 chromosomes means 23 pairs, 22 pairs of autosomes and one pair of sex chromosomes. Okay, so autosomes, these are non-sex chromosomes. We have 22 pairs. Okay, and sex chromosomes, they will determine the sex. There is one pair for it and they are labeled as X and Y. Okay, this pair is labeled as X and Y. Then these autosomes are from chromosome 1 to chromosome 22. That is how they are labeled. Okay. Now let's see the types of chromosome based on the centromere. This we have seen that these chromatids are attached at the level of the centromere. Now, depending upon the position of the centromere, it may be metacentric, submetacentric, acrocentric, or telocentric. Okay, so these are the types of chromosomes. However, telocentric chromosomes are not seen in the human beings. Okay, so human beings, we have only metacentric, submetacentric, acrocentric. So that can be asked in the SBA. So depending upon the centromere location, we have metacentric where the position of the centromere is in the middle. Okay, so you get two equal arms. So this is present in the human. Then you have submetacentric, that is centromere position is slightly off the center. You get two unequal arms. This is also seen in the human being. Then next you have an acrocentric where this centromere is more towards the end. Okay, so it is near one end and very short. P arm is very short here. Okay, this is also seen in the human being. So which one is not seen in the human being is the telocentric, where this centromere is exactly located at the end of the chromosome. Okay, so this is not seen in the human being. Only one arm is there. So this is not present. Okay, there is another classification. There are two types of classification systems. Okay, one is called as Denver's classification, where these 23 pairs, they are grouped into A to G groups are made. Okay, so it is a morphological classification. Another classification is on the banding of these chromosomes. It is a detailed classification, that is the modern classification which is accepted. Okay, so on this there are generally no questions are asked, but this depending upon centromere location you should know. Okay, so these chromosomes are divided by this Denver classification into A to G groups, and chromosome 1 to 3 are in A group, 4, 5 are in B group. Okay, so that is how these chromosomes are divided in the Denver classification. Okay, and there is a difference between the Denver and Paris. This is just for the information. Okay, so basis for the Denver's classification is the size of the chromosome and the centromere, while it is the banding pattern which is the basis for Paris classification. The accuracy is basic for the Denver, while it is advanced for the Paris classification. Denver is the older system, while current system which is followed is the Paris system. Okay, so abnormality detection limited role for the Denver, while it is precise in the Paris classification. So in Paris, the basic idea is the advanced system using chromosome banding technique, that you should remember. Here the chromosomes are classified as per the banding technique. Okay, so basis, it uses staining methods like G banding, Q banding, so it produces light and dark bands. Okay, so what it tells us, precise identification of the chromosomal regions and the structural abnormalities. Okay, like 46XX, 47XY plus 21, 46XX deletion 5P. Okay, deletion 5P, where do you see? We will see this, I think, in cry-du-chat syndrome, you get deletion of 5. Okay, so acrocentric chromosome, you remember, it is involved in Robertsonian translocation and Down syndrome. This you remember, acrocentric chromosomes, where they are involved. They are involved in the Robertsonian translocation and the Down syndrome. You may come across this in the SBA questions. Okay. So let us see about the molecular organization of these chromosomes. So how these DNAs are packed. So DNAs are packed into histones. They form the nucleosomes. Then there is a chromatin and there is a chromosome. Okay, so when this DNA is a huge material and then it is packed into the histones, these are the subtypes of this. Okay, so the final unit is the chromosome. Okay, so DNA in our body is about 2 meters long, so it must be tightly packed. So first DNA wraps around the histone proteins, forming nucleosome, which looks like a beads on a string. These nucleosomes coil further to form the chromatid. Okay, so what happens? First, DNA wraps around the and histone proteins, and it forms the nucleosome, which looks like a beads on a string. These nucleosomes will coil further to form the chromatin, and finally the chromatin will condense into the chromosomes during the cell division. Okay, so what is nucleosome? It is the DNA wrapped around the histone proteins, is called as nucleosome. Okay, so appearance is like a beads on a string. So it is a function is it is a compact DNA, regulate the gene expression. So euchromatin versus heterochromatin. Euchromatin is the active form of chromatin, while heterochromatin is the inactive form of chromatin. In euchromatin, it is loosely packed and generally genes are expressed here. While in heterochromatin, it is inactive, it is densely packed, and there is a mainly structural role for this heterochromatin. So what are the functions of chromosome? It is the genetic information storage. They will carry the gene and will determine the traits. Second, transmission of heredity. They are passing the information from parents to the offsprings. Third is a cell division. Ensure accurate DNA distribution during mitosis and meiosis. Next is a regulation of gene expression. Chromatin structure controls transcription. Protection of genetic material. So telomeres prevent the DNA damage. This is the most important. The function of the telomere is to prevent the DNA damage. This also you should remember from the SBA point of view. Okay. Then clinical correlation. So telomere dysfunction. That means the end portions, if they are defective, what it will lead to? One is aging and second is a cancer. Okay. So aging or apoptosis can happen physiologically, while if there is some pathological problem, it may lead to the cancer. Then centromere error can lead to non-disjunction, that will lead to the aneuploidy. Example we have seen, either there can be trisomy 21 or monosomy X, that is Turner syndrome. Okay. Then there can be structural abnormalities. Deletion will lead to cry-du-chat. Okay. 5P deletion we have just mentioned few seconds back. Okay, then there can be translocation. Translocation is seen in Down syndrome. Duplication can lead. Duplication is a used in the gene amplification. Okay, so important point to remember is a centromere is the site of spindle attachment. Telomere are the protective ends. Euchromatin is the active DNA. Heterochromatin is the inactive DNA. Acrocentric chromosomes, there is a risk of translocation. Nucleosome is the DNA plus histone unit. Okay, next.
Basic unit of DNA package is histone, sir. Option D.
It should be, I think, nucleosome or histone. Let us see. Yeah. See, nucleosome. Okay, nucleosome is the DNA wrapped around the histone protein. So DNA which is wrapped around the histone becomes nucleosome. Okay, next.
Which chromatin is transcriptionally active? Sir, euchromatin. Option B.
Correct, correct. It is the euchromatin which is active. Next.
Which chromosome type is not present in a human? Sir, option D. Telocentric.
Correct, correct. Next.
Which chromosomes are acrocentric in humans? Sir,
Acrocentric
13 is
chrome.
there. 13 is there. So option D. That table.
Just 13, 14, 15, 21, 22. Okay. Acrocentric, you remember, please, because they are concerned with the translocation also. Okay. What is the main function of telomeres?
Main function of telomeres. Option A, DNA replication, protein synthesis, protects the chromosome ends, spindle attachment, and gene transcription. So protects the chromosome ends.
Correct, correct. So telomeres prevent DNA degradation and instability. Next question.
Why is X inactivation important? Prevents mutation, reduces chromosome number, ensures dosage compensation, increases gene expression, activates Y chromosome. Inactivation is important. Reduces chromosome, increases gene, prevents mutation. Sir, option D, increases gene expression.
No, it is
It
option
prevents
C.
overexpression.
It ensures the dosage compensation because male and female should have the equal dose, no?
Okay,
It
yes.
ensures the dosage compensation. Okay, so it is C. Balances the gene expression between XX females and XY males. I have explained you at that time because there is XY, only one X. Okay, that X will carry certain information for certain genes. So they will have, suppose they are n number of times a particular protein is synthesized, but that protein should be equal in quantity in n number of times only. Otherwise, in females, it will be 2n. Okay, which is coded because of 2X chromosomes. This should not happen.
Which state? Okay, sir. Which statement about Y chromosome is correct? Gene rich, contains many housekeeping genes, contains SRY gene equal to X chromosome present in females. Sir, contains SRY gene.
Yes, it contains SRY gene. Y chromosome is gene poor, okay, but main function is via SRY gene. Next question.
X-linked diseases are more common in males because males have two X chromosomes. X chromosome is inactive. Males have only one chromosome. Y chromosome is large. Female lacks X chromosome. X chromosome is male have only one X chromosome. Yes, sir. Option C, males have only
Yes,
one X.
very straightforward. Males have only one chromosome. Males are hemizygous, so no backup copy of X gene. Okay, so just take home message. Bar body is inactive X chromosome. Gist is leads to the inactivation. This is the gist gene. Okay, then telocentric chromosomes are absent in human being. Acrocentric numbers you should remember. Okay, mosaicism definition. Okay, two different cell lines are produced. Okay, in same species. That is the mosaicism.